Welcome to this explainer. It's wild to think about, but there is a high-stakes, incredibly intense world existing right under our noses, and a lot of the time right inside our bodies. Have you ever actually thought of doctors, microbiologists, and lab techs as detectives? Because honestly, that's exactly what they are. Every single day, they are treating the human body as a literal crime scene, trying to identify the exact microscopic culprits responsible for infectious diseases. So today, we're going to unpack the principles of diagnosing microbial infections and completely demystify how these medical detectives actually crack the case.
Picture this. You wake up, your fever's spiking, you're exhausted, everything hurts. How do doctors actually figure out what's causing it? Is it a rogue bacterium, some stealthy virus? Without knowing exactly who the invader is, doctors can't prescribe the right weapon, like the right antibiotic or antiviral to help you heal. Solving this mystery takes a ton of precision, a lot of patience, and some seriously amazing forensic science.
So, what exactly is the crime here? A microbial infection isn't just a germ hanging around. It's an actual disease that comes from a dynamic, ongoing clash between a host, that's you, and an infectious agent. And who wins that clash depends on two main things. First, the microbe's virulence, which is basically how aggressive it is. And second, your immune status. If a patient is already dealing with an underlying illness or they're on immunosuppressive drugs, their defenses are down, giving the microscopic suspect a major upper hand.
All right, let's get into phase one, the microbial suspects. Because to solve any crime, you first need to know who you're looking for, right?
Check out the usual suspects. Notice the crazy difference in scale and structure here between our two most common offenders? On one hand, you've got bacteria. These are single-celled organisms that can multiply super fast all on their own inside your body. On the other hand, you've got viruses, which are essentially just tiny packages of genetic material wrapped up in a protein shell. Viruses are the ultimate freeloaders. They literally cannot survive or multiply on their own. They have to hijack your cells to do their dirty work. Because their structures are so different, the tools we use to catch them have to be completely different too.
But wait, the lineup gets weirder. Bacteria and viruses aren't the only ones causing trouble. We've also got incredibly diverse threats like fungi, which can be anything from microscopic yeasts to larger molds. And then there are blood-borne parasites and intestinal worms. These guys operate by totally different rules. Trying to catch a yeast infection like Candida takes a completely different investigative strategy than hunting down a parasite swimming in the bloodstream.
A massive part of the investigation is figuring out where these suspects even come from. Sometimes the call is coming from inside the house. We call these endogenous sources. Basically, your normal body flora just going rogue. E.coli, for instance, is totally fine in your gut, but if it wanders into the urinary tract, boom, infection. Same with MRSA, which can just chill peacefully in your nose until it causes a severe wound infection. Other times, the threat is exogenous, coming from outside stuff, like bad water or soil. Then you've got zoonosis, diseases jumping from animals to us, like Ebola or SARS-CoV-2. And obviously, you can just catch things directly from other infected people.
Oh, and we definitely can't forget the getaway drivers of the microbial underworld, insect vectors. Some parasites simply can't move from host to host on their own. Take the Plasmodium parasite that causes malaria, for example. It uses the female Haemophilus mosquito as a literal getaway vehicle to get into your bloodstream. So spotting the vector is often a huge clue for medical detectives trying to figure out what a patient has contracted.
Now that we know who we're up against, let's move to phase two, starting the investigation. So how do we actually gather the evidence? The golden rule in microbiology labs is MCS: microscopy, culture, and sensitivity. Step one is collection. You have to get a really high-quality specimen: blood, urine, cerebrospinal fluid, whatever it is. And here is a totally crucial rule for any crime scene: gather the evidence before it's destroyed. For doctors, that means getting the sample before giving the patient any antibiotics, which could wipe out the microbes before the lab even gets a look at them. Step two is culturing, to grow the evidence. And step three is testing its sensitivity to treatments. Let's look at how that actually works.
All right, let's step out of the clinic and head into phase three, inside the diagnostic lab. This is where the real forensic magic happens.
Going back to that rule about protecting the crime scene, the absolute biggest challenge when first plating a specimen is avoiding contamination. Remember that endogenous skin flora we talked about? Well, if a lab tech accidentally drops a tiny bit of normal skin bacteria into the sample, it's going to grow in the lab and totally hijack the investigation. A contaminated sample means a false diagnosis, so literally everything has to be meticulously, perfectly sterile.
Once it's safely plated, scientists have to coax the microscopic suspects out of hiding. A patient sample might only have a few individual microbes in it, right? So we have to grow them into the millions just to see them. We do this by feeding them the perfect, nutrient-rich diet using artificial media, stuff like nutrient agar, blood agar, or MacConkey agar. Given the right environment, the bacteria multiply rapidly into these visible, vivid colonies. They finally reveal themselves.
But just looking at a blob of bacteria isn't enough. We need a fingerprint. And that brings us to chemical staining. Think of this exactly like a detective dusting for prints. By adding specific chemical dyes, technicians can tell microbes apart based on their cellular structure. The classic Gram stain turns certain bacteria purple and others pink or red. Another really vital one is the Ziehl-Neelsen or ZN stain. It catches what we call acid-fast bacilli, which is the exact fingerprint we look for to catch incredibly stubborn pathogens like tuberculosis.
We don't just look at how they're structured, though. We test their behavior. Biochemical tests help nail down the suspect's exact identity by looking at their diet. Scientists put the bacteria into different tubes with specific sugars to see what happens. Do they ferment lactose or sucrose? Do they produce specific enzymes like catalase or coagulase? If they do, the liquid in the tubes actually changes color. It is basically a chemical confession telling us the exact species we're dealing with.
But what happens when the bug is just a master of evasion? Or what if it's something super deadly, like Ebola, that's just way too dangerous to culture normally? That brings us to Phase 4, Advanced Detection Techniques.
If we can't easily find the microbe itself, we look for the footprints it left behind in the immune system. I'm talking about antibodies. When you get infected, your body creates specific antibodies tailored to fight that exact bug. Testing the blood for these tells an amazing story. If we see IgM antibodies, which usually show up in 1 to 10 days, we know there's an active, acute infection happening right now. But if we find IgG antibodies, which take 2 to 4 weeks or more to show up, that points to a chronic infection, a past exposure, or even a successful vaccination. It's an internal timeline that proves exactly when the body was attacked.
And when we need absolute 100% certainty, we break out the big guns, molecular fingerprinting. Techniques like PCR, real-time PCR, and nucleotide sequencing are the absolute cutting edge here. Instead of waiting days for bugs to grow in a dish, scientists can extract the actual DNA or RNA of the pathogen directly from the sample and amplify it. It is incredibly fast and astonishingly accurate because it identifies the pathogen's literal genetic code.
Okay, we've identified the suspect, but a detective's job is not done until the threat is actually neutralized. Let's move to Phase 5. Solving the medical case. So how do doctors know exactly which weapon will take down the microbe? They use antibiotic sensitivity testing, specifically something called the disc diffusion method. They literally take a plate covered in the bacterial suspect and drop tiny paper discs on it, each one soaked in a different antibiotic. After letting it sit, they check for zones of inhibition, these clear, empty rings around the discs where the drug successfully killed the bacteria. It's direct, visual proof of what will save the patient. No guessing, just hard evidence. But here's the catch with all this incredible science. It takes time. And waiting for test results when you feel awful is the worst, right? But look at the timeline. A standard culture needs at least 18 to 24 hours just to grow. Finding out which antibiotic works takes a minimum of 48 hours. Blood cultures take 3 to 5 days just for preliminary results. And if we're hunting tricky suspects like fungi or tuberculosis, you might be waiting 2 full weeks or more. Medical detectives literally have to have this time. Biology simply cannot be rushed, and rushing means you might miss the real culprit.
And that wraps up our investigation. We've seen how careful collection, culturing, staining, and genetic testing let doctors pinpoint infections and save lives every single day. But here's the crazy part. This is an endless arms race. Microbes are constantly mutating, finding new ways to resist antibiotics and dodge our tests. So I'll leave you with this thought. As these microscopic bad guys get smarter, what kind of wild, cutting-edge tools are our medical detectives gonna have to invent next just to stay one step ahead? Thanks so much for hanging out with me in this explainer. Keep wondering, keep learning, and I'll catch you next time.